And obviously, when hydroperoxide levels are very high, there has to be a threshold. When they get to beyond that threshold, oxidation will damage the DNA, not due to rupture, but due to the creation of other pathways, and genomic instability may also be caused, as well as apoptosis. The induction of apoptosis in this sense means eliminating a cell that had increased its potential enormously. But that's not all, because activation by oxidation-driven redox switch will also control very important molecules in physiology and pathology. For example, HIF, and therefore angiogenesis, and the metabolic shift typical of metabolic syndrome. HIF controls angiogenesis, and this raised the interest of oncologists. It also controls PI3K kinase, that is the cell's ability to grow and proliferate, the ERK kinase pathway for proliferation, also the NFKB pathway for inflammation, and last but not least, it also controls the activity and function of the precursor of all oncogenes, CSAC, which is activated in many mechanisms related to malignant biological activity. So, to sum up, the substances that combat and that counteract peroxidase signaling can be summed up as effects that we've described. I'll look at the bottom first. Xenohormetic effects, that is, molecules that defended the plant can now defend us, and therefore they inhibit the production of peroxidase, they can also optimize in various ways the coupling of the mitochondrial respiratory chain, and they can also control lipooxygenase. And that's very important because lipooxygenase is an element where plant and animal overlap is very significant. And I'd also make another comment here. We're talking about nutrition, that is the hormetic effects. The hormetic effects are those described by Paracelsus in antiquity, and something that is bad in high doses might not be less harmful in low doses. In fact, at low doses it may be helpful. This brings us into a number of very interesting molecules which are on the list of things that are harmful, but being on the list of things that are harmful, they also do something that's biologically useful. They activate defense mechanisms. We'll be hearing more about this from our colleague. CHIP1 NF1-2 pathway is the one that's involved, and a very large proportion of the prevention of neoplastic cell behavior is caused by the induction of these mechanisms, these systems. In other words, what we have to do is not so much beware of the harmful ones, but we need to place a great deal of attention on not overdoing it, if you will. For example, one of the major inducers of defense mechanisms are hydroxynonenals, and these are defined as some of the toxins in chips, potato chips. However, science would suggest that inducing NFR2, which you can obtain in hurry, curcuma, broccoli, cabbage, etc., you get the same effect from potato chips. So perhaps a potato chip once in a while, as we were saying before about the goodies and baddies, might not be a bad thing, insofar as it induces defense mechanisms. So, to close on this part of my lecture, I'd like to show you just one slide from one of our own studies. I hope you can see it clearly enough. And this is NAC, which, as you can see in A, these are neoplastic cells, I think, cancer cells. These are localized in the membrane, where they activate all the various bad things that we can recognize. So, cell proliferation, detachment from the matrix, the ability to metastasize, etc., etc. So treatment with the peroxidase reducer, incorrectly called antioxidant, very quickly can shift CSARC towards the endosomes where it is degraded. And I'll add that under these conditions, we will slow down and almost block proliferation and activate cell differentiation. And this is precisely what we want to control malignant neoplastic behavior. So what are my conclusions? Essentially, we can say that plant-derived phenolic compounds can play a protective antioxidant effect only on the skin, where there are lots of them, and the GI tract. Secondly, plant phenolic compounds are apparently competent for controlling damaging stress response, and this obviously has nothing to do with the antioxidant effect, and it can be obtained with the right concentrations. There are some non-free radical scavenger neophilic antioxidants that seem to be relevant in controlling redox switches, the osteoinductive mechanisms, and also for controlling cellular differentiation, proliferation, and survival, which are all very important in malignant growth, cell development. So this is the lady saying to her caveman friend, my doctor says you should be drawing more fruits and vegetables. Well, it's been given a modern twist, and I'm sure that is an aspect that will be studied in future. Thank you.
I would like to thank Professor Ursini for his extremely clear and exhaustive presentation. Now, I think that we can leave questions or discussions to the end of this session. I would now like to give the floor to Professor Umberto Cornelli from the Loyola University of Chicago, who will speak to us about a subject that's correlated to this, which is the role of antioxidating molecules in dealing with oxidative stress, which is very important from the biochemical point of view.